Well, hello everyone. Welcome to another episode of STEM Nerds. I'm Jason. Glad you're here. In today's video, we're going to be talking a little bit about microbial ecology because we talk about microbes a lot on this channel, and one of the things that we do is we tend to focus on some of the bad things that microbes do, and sometimes we forget that microbes are absolutely essential for the environment and for ecology in general. And so in today's video, we're going to start a discussion on just how important those microbes are. All right, so here we have the trophic levels, the food pyramid, if you will, the food web, and we tend to look at this on a macro scale where we have our photosynthetic plants and grasses and trees and bushes and things like that, and those are the primary producers, and then of course those things get eaten by primary consumers, and we work our way up the chain to secondary, tertiary, quaternary, and quinary types of consumers. But in microbiology, our focus really tends to be towards the bottom of this energy or food pyramid, and that is the photosynthetic organisms. We tend to sometimes forget about things like cyanobacteria, algae, and these very small microbes that are also photosynthetic, and when you combine them all together, they actually fix more carbon, release more oxygen than all the land plants combined. But not only that, at the beginning, the base of that food pyramid, not only do we have the primary producers, which are often microbial in nature, microscopic, but we also have heterotrophs, that is our chemo-organoheterotrophs, that are microbial in nature as well, and they can be consuming these primary producers. Not only that, but the decomposers are really, really important in this food web, and that would consist of your bacteria and your fungi breaking down things that other organisms would not consume. And so the point here is, on a microbial side, microbes are absolutely critical when it comes to overall health of ecology and ecological system and the food web. This is a cool little thing that we do sometimes in microbiology, and that is set up this thing called the Winogradsky column. And what you do is you basically pack the thing full of mud, you put some water on top, you seal it, and then you cover the bottom parts with foil and stick it in front of a window. And in about a couple months, you have a really interesting experiment. And what it does, as you can see here, is it shows you where microbes naturally inhabit certain parts of an ecosystem, or we can take a closer look at soil microbes. And so at the top, where you're getting a lot of sunlight and a lot of oxygen, you're going to have things like algae and cyanobacteria. Then just underneath that, you're going to start seeing more of your anoxygenic phototrophs, something that we talked about in a previous video. I'll put that in the upper right-hand corner as well as in the description below. But our anoxygenic sulfur and non-sulfur photosynthetic microbes are really important because they still need visible light, but now they're taking some of those waste products that they're finding in the soil and other metabolic products, and they're utilizing those for an electron source. And as we travel further down, we start seeing a lot more anaerobes and a lot more chemolithotrophs. And again, it's just a kind of an interesting experiment to do to see microbial ecology play out in real life. So our focus today, very briefly, is going to be on two different types of ecosystems. One, aquatic, and then the second one, soil, because microbes are really important for both. And so when it comes to aquatic ecosystems, the picture that you're looking at here is your typical ocean water. And based on what we already know about microbes and how we classify them, well, we can classify the typical microbe that we might see in ocean water. One, they're usually going to be at least halotolerant, but most likely halophilic. Halo meaning salt, phile means to love, and so these are going to be salt-loving microbes that are going to enjoy that roughly 3.5% sodium chloride found in most ocean water. The next thing is, well, temperature. Most of these microbes, depending on where they're at in the world, could be anywhere from psychrotrophs that are going to enjoy cooler water all the way up to mesophiles, especially as you get into warmer ocean water. In terms of what they're going to be using for a terminal electron acceptor, depends on where you find them in the water column. The closer to the surface they are, the more flow you have in the water, the more aerobic these microbes are going to be. But as you go further down in the water column, and especially as you travel into the substrate in the ocean, you're going to see a lot more anaerobic microbes. And of course, depending on where you're at in the water column, you could see phototrophs, again, especially as you get closer to the surface. And the clearer the water is, you might tend to see more phototrophs. Just remember, they have to have access to visible light. And so as you go down in the water column and you lose that visible light, you're going to be seeing a lot more chemotrophs. And certainly as you get into the substrate, you're going to see a mix of chemoorganotrophs as well as chemolithotrophs. pH of ocean water is generally going to be a little bit above neutral, but you're usually going to find some neutrophiles there, maybe some chalophiles depending on where you're at, but for the most part, you're going to see a lot of neutrophiles. In a freshwater habitat, really the only thing that's going to dramatically change is the fact that you're not going to see halophiles there anymore. You're going to see non-halophilic organisms, possibly some halotolerant, but everything else is going to be the same based on where you find the microbes. Again, the closer you are to the surface, the more aerobic, the more chemoorganotrophs. The closer you are to the surface, you're going to see more phototrophs. As you go down in the water column, you might start to see more anaerobic organisms, and by the time you get into the substrate, a lot more anaerobic organisms, a lot more chemotrophs, a lot less phototrophs. And one of the things that's really interesting about the aquatic ecosystems that we're looking at is the water can have varying amounts of clarity. Now, normally, most water on Earth, and certainly ocean water, is going to be oligotrophic. And what that means is the water is going to be clear, but it's also going to be relatively nutrient poor. That's important because microbes in this area, they need things to grow. They need space, proper temperature and environmental parameters, and they need food in the form of nutrients. And often in aquatic environments, it's the nutrients that are a limiting factor. In eutrophic waters, that tends to be a lot more nutrient rich. You tend to get slightly cloudier or more turbid waters as a result. And sometimes we screw up our aquatic ecosystems, and it's initially the microbes that make the changes and the adjustments. And so I want to spend a few minutes about how that can happen because it's a really important point, not just of microbial ecology, but ecology in general. And so one of the things that we study in the sciences are these areas called dead zones. And these dead zones are basically areas in the ocean where there is not a whole lot of life because we've switched from oligotrophic to eutrophic waters. What's interesting is where it starts is often very far away from an ocean or from a shore. It can actually start in the middle of a country, and it really begins when you add fertilizers to soils as part of an agricultural process. From there, if we are not properly managing the amount of nutrients we're adding to the soil, and we combine that with some runoff due to a lot of rain or possibly over irrigation, those excessive nutrients can run off into local streams and rivers. From there, those excessive nutrients can run off into the Mississippi River. We're going to use an example here in the United States. And so when those local streams and rivers dump off those extra nitrates and ammonia and nitrogen-based fertilizers and sulfur and phosphorus, that can wind up in the Mississippi River and eventually make its way into the Gulf of Mexico. We're going to have low amounts of nutrients, low concentration of nutrients, but as you might expect in the Gulf of Mexico, you're going to have plenty of sunlight, you're going to have plenty of space, you're going to have really consistent environmental parameters. And so that's great for bacteria, but the thing that is preventing them from really growing is the fact that there aren't enough nutrients to make them go crazy. Well, when we dump a lot of nutrients in the soil and it eventually runs off into local streams and rivers, then into the Mississippi, then into the Gulf, guess what we've done? We've converted those oligotrophic waters into eutrophic waters. Well, we took that limiting factor away, and now we start to see changes, but those changes begin on the microbial side. So now that we have converted those oligotrophic waters to eutrophic waters, what we've done is we've added a lot more nutrients, and that is going to provide the nutrition for phototrophs to flourish. So we get this initial increase in phototrophs because now they've got all this food, they have the sunlight, they have the space, and as they increase, a couple things happen. They will increase in concentrations, and then of course they're going to die off, but they're also going to be consumed by aerobic chemoorganotrophs. And again, this starts out microbial in nature, but there's a problem here. And that is those aerobic organisms start consuming a lot of oxygen as a byproduct of their metabolism, something that we talked about when we discussed the electron transport chain. Once they start to increase and remove a lot of the available oxygen, a lot of the larger organisms in those waterways, they can't escape. These dead zones, these hypoxic areas, can be hundreds, if not thousands of square miles. And so you've got slower moving organisms like a lot of your crustaceans, your smaller fish, they just can't get out of that area. And so they essentially suffocate and they die. And of course, now they are organic compounds that can be used by more aerobic chemoorganotrophs or even anaerobic chemoorganotrophs. And at this point, you have a growing issue. These dead zones will fluctuate in size depending on the season, and they are not just in the Gulf of Mexico, they are in many other parts of the world. But again, the point here is that it all started with us adding nutrients to soil, and it really started as a microbial adjustment in the ecological system that eventually impacts larger organisms that we can actually see. Well, there's not just your standard freshwater and saltwater ecosystems. There are lots of other types of ecosystems that are aquatic that are actually pretty extreme. Things like thermal ponds, and you've got these areas where there's a lot of sulfur, like the sulfur springs, where they can be really, really acidic and really hot. Yellowstone National Park actually has a walkway where you can go through these really, really hot waterways, and you have to be careful because if you were to actually go into those things, you might not make it out, and that happens every once in a while. And so what we have here are really not a whole lot of macroorganisms, but there's lots of microbes, and that's the reason for the colors that you see here in this picture. You're going to have a lot of archaebacteria, extremophiles, so extreme thermophiles, halophiles, acidophiles. These waters can be extremely acidic. And so you have this entire microbial ecosystem when really that's the only thing that can live in these areas. Another really great example of an extreme aquatic environment are those that are extremely high in salt. This is the Dead Sea. Up to 30% sodium chloride. That is way too much for most macroorganisms to survive. But the microbes, again, are extreme halophiles. They can do this. They have a way to balance out osmotic pressure on either side of their membrane so that they can survive in these concentrations of salt. They can get up to 30% sodium chloride, which is absolutely amazing. I want to switch gears here for a couple minutes because there is another ecosystem we haven't yet discussed that is really important in microbiology, and that is the soil. The soil could potentially hold hundreds, if not thousands, of antibiotics and antimicrobials. It is a flourishing ecosystem that we really don't know that much about because many of the microbes that grow in soil, we just cannot grow in a lab. And so a lot of the information that we get comes from genetic studies, something that we'll talk about in a little bit. So there could be hundreds of antibiotic-producing microbes, and there are hundreds. There could be hundreds, if not thousands more, because in nature, what these microbes are doing is they're using antibiotics to control the growth of their neighbors and their potential competitors. What we do as human beings is we isolate these substances, we ensure their safety, we put them into human beings, and they wind up killing microbes that cause infectious disease in humans. There are also symbiotic relationships that, quite frankly, we don't fully understand. These symbiotic relationships are often between bacteria and plants and fungi and plants. And what we're doing more and more is as we understand these relationships, we can then rely less on those fertilizers that are causing all of those issues I just talked about and can be expensive, both in terms of manufacture, distribution, and then, of course, the cleanup if they are misused. But if we can understand the microbes in these ecosystems, it's possible that maybe we can add microbes to the soil that do a lot of the work that fertilizers normally do in a symbiotic relationship with plants, as we know they already do. And so the microbes will provide plants with nitrogen-based compounds, while the plants at the same time provide the microbes with sugar-based compounds. The other interesting thing about the soil is there are a lot of spore-forming microbes. As you can imagine, in large parts of the world, environmental conditions can change pretty dramatically from one season to the next. Well, how are the microbes how are they going to deal with that? They don't just all die. They often will form spores, something that we've talked about numerous times before. And these spores allow the microbes to withstand extreme environmental parameters. So desiccation, drying out, which can happen in certain parts of the season, changes in temperature, lack of food, lack of moisture, as we already talked about. Now, the downside to these spore-forming microbes is the fact that if they are potential human pathogens, they can be really difficult to get rid of because they're resistant to UV. They're resistant to antibiotics because they're really not growing at that time. They're in a metabolic state that is inactive. And so we have to be really careful around some of these spore formers that cause human disease. All right, so the last thing I want to cover in this video is really thinking about animals as ecosystems for microbes. It's really easy to think about the outside world, but as it turns out, human beings, animals in general, are important ecosystems and something worth considering. I want to really focus on three main types of animals, and I want to focus, at least for right now, on one part of those animals, and that is the digestive system of animals, because it is so critical that the animal and the microbe form that symbiotic relationship. So let's take a closer look. So our focus here is going to be on three different types of animals. We've got a carnivore on the left-hand side, and then in the middle, on the right-hand side, we have two different types of herbivores. And the reason I want to focus on these three organisms is the digestive systems are very different. In the middle, we've got a rabbit. On the right-hand side, we've got some type of ruminant organism. And we're going to see, at least in the middle and on the right-hand side, the microbes are absolutely critical. And in fact, if they were not present, we would have serious ecological issues. All right, so let's start here with the rabbit. The rabbit primarily consumes herbivorous materials. And what's interesting about it is without microbes in the rabbit gut, it probably wouldn't be able to survive. Let's take a closer look. So here we have a picture of a typical rabbit digestive system, and you're going to see there's a lot of similarities to us. We have the stomach, and we have the small intestine, and then we have this big green part called the cecum, and then we have the blue area called the large intestine. Now, for those of you who are familiar with the digestive system of mammals in general, we know that we eat food, it goes down our esophagus, into our stomach, and we have some digestion going on there, and then into the small intestine where most of the nutrient absorption occurs, into the large intestine where we're reclaiming water, some vitamins, and then eventually it's eliminated from the body. Okay, so that is the case with most mammals. But there's a problem. Rabbits eat a lot of grass. That grass contains high concentrations of cellulose, and we do not possess the enzymes necessary to break down cellulose. And in fact, the rabbits don't make those enzymes either. And so what winds up happening are those grasses wind up in the stomach. They go to the small intestine where there is some small amount of nutrient absorption, but the overwhelming majority of the organic compounds, and in fact the energy that the rabbit needs, passes through the small intestine and then goes to this green area here called the cecum. That is where microbes do their thing. There are microbes there that can break down the cellulose into individual sugar molecules. Problem is that food can't go backwards back into the small intestine where nutrient absorption normally happens. It has to go through the large intestine. And so the way that a rabbit often gets around this is it passes that food through the large intestine, it exits out of the body, and yes, it turns around and eats its waste material. Well, why did the rabbit do that? It did that because at this point, all of those carbohydrates are now in a form that are readily absorbable. So it has to re-eat that, and then when it goes back through the small intestine, now we have those monomers or the disaccharides that can be easily absorbed, and we're not worried about the cellulose. And so if you see a rabbit eating its own fecal matter, it's doing that because it's getting the vast majority of the nutrients it needs from that, not from the initial grass that it eats. All right, so that was kind of gross. I get it. I know that. Gonna have to be honest with you, the cow is not a whole lot better when it comes to how it consumes its nutrients. Let's take a closer look at the cow, the cattle, the organisms that have a rumen. All right, here is your typical cow digestive system, and the arrows kind of show you what's going on. Cattle will eat grasses all day long, and those grasses, again, cellulose-based materials, will travel into the rumen. Think of that as a big, giant, almost like an incubator, where there are microbes that, just like in the cecum of the rabbit, those microbes produce enzymes that can break the cellulose down. Now, the problem is the rumen does not have a direct connection to the small intestine of the cattle. And so what they have to do is they regurgitate what they've broken down, and then they swallow it again into what is known as their true stomach, where it can eventually go to the small intestine, where you get nutrient absorption, and then to the large intestine, and then eventually out. And so a cow, unlike a rabbit, doesn't have to re-eat its own fecal material, but in a sense, it has to re-eat its regurgitated food that it ate earlier in the day. And in both cases, and the reason why we're talking about it here is for the organisms that are eating primarily grasses, cellulose-based materials, they wouldn't really be able to get much out of that if it wasn't for the microbes that were able to break down those bonds in the cellulose and freeing up those individual carbohydrates for nutrient absorption. Now, I mentioned this briefly earlier in the video, but it turns out so many of the microbes that we know to exist in systems, ecological systems, we simply cannot grow them in a lab. And in fact, we can probably only grow less than 1% of the microbes we know to exist. And so how do we know that they are there when they're forming these very intricate, very complicated ecosystems, not only with the organisms they rely upon, like we just talked about, but with each other? That is where metagenomics comes in. And in metagenomics, what we're doing is we are taking samples from a particular ecosystem, and we are genetically sequencing those samples to determine what microbes are present. And so once we have the DNA sequences, we can link those to microbes that are present. And in fact, we can actually do quantitative sequencing where we can get an idea of not only the microbes that are present, but in what quantity relative to one another. And this has been hugely important in studying soil microbiology, the digestive systems of animals, and the digestive systems of human beings as well. So that was a very brief look at microbial ecology. As always, thank you so much for being here. Have a great week, everyone.